Okay, so hello everyone. We're going to be using this video as a way to go over some of the key concepts that you'll need to be familiar with to be able to do the DNA analysis task, which counts for 15% of your grade. And this task, it should hopefully be accessible to everybody. So even if you are only just starting to learn how to code, we've tried to design it in a way that you will still be able to get something out of this. And if you are a little bit more experienced with coding, hopefully you'll be able to have a little bit more freedom and creativity with this task as well. So before we get into it, I think we should have a bit of a background on DNA sequencing in general. So this is some content that you may have encountered before in other modules, but don't worry if not, we're going to go over the basics now. So in essence, there are three different generations of sequencing that are currently available in market. We have first generation Sanger sequencing, second generation or next generation sequencing, and we also have third generation sequencing, which is long read sequencing, which involves or is sort of provided by companies like Oxford Nanopore and PacBio. We're not really going to talk too much about third generation sequencing. We're just going to be focusing now on first and second generation sequencing. So the task that you will have to do will be to do with some simulated NGS data. But in order to understand NGS, we need to first understand where it came from, which is Sanger sequencing. So I think with all sort of sequencing, we do this because we want to know what our DNA sequence is within a target region or a target DNA molecule. And sequencing begins in terms of Sanger chain termination from a defined starting point. So we start with a DNA primer. So this is a 20 nucleotide oligo that has homology to our target sequence. And Sanger chain termination is based on the polymerase chain reaction, or PCR. So like any other polymerase chain reaction, we're going to be supplying, our polymerase, our workhorse, whatever sort of buffers required for this reaction to take place, as well as a mixture of dNTPs. Critically, we also have in this mix some fluorescently labeled ddNTPs. So during this reaction, we have extension of our DNA molecule and a random incorporation of both normal dNTPs as well as fluorescently labeled ddNTPs. And extension of these molecules terminates upon addition of a fluorescently labeled ddNTP. So each ddNTP, so a G, an A, a T, or a C, all fluoresce a different color, and this is how we identify which base has been incorporated. So all of these different fragments that have terminated at different nucleotides, they are different sizes. And when these are ordered from large to small fragments, we're able to ascertain information about the sequence of the DNA. So we fire a laser at all of these different fragments on a gel, and they fluoresce different colors depending on what that terminal nucleotide is. And when you read this information out, going from the smallest to the largest, we end up with information on the sequence. So, and this kind of information can be represented and often is represented in a chromatogram. So something that looks a little bit like this, where each peak is the level of fluorescence detected at a particular wavelength. So the wavelength that is associated with the fluorescence of that base. So for those of you who will be doing sort of lab-based third-year projects, you will most likely encounter Sanger sequencing if you're going to be doing some cell and molecular biology, where if you need to clone a plasmid, if you need to generate a plasmid, Sanger sequencing is probably the most cost-efficient and sort of time-efficient way to verify that what you've added to your plasmid and added to and cloned it to your plasmid, I think is the terminology we use in the lab. We most often verify this by Sanger sequencing. So now I was planning to play a video, but I think we're going to sort of leave that and I'll link to that somewhere on the Canvas page. So if you wanted to watch the sort of manicured and beautiful video explaining next generation sequencing from Illumina, which is the main provider, it's available for you to watch. But let's just break down sort of the basics that you need to understand. So the thing that sets apart Sanger sequencing and next generation sequencing is, first of all, it's incredibly accurate, so high levels of accuracy with each base that is identified and called by next generation sequencing. Also, we get a wealth of information that you don't often get with Sanger sequencing. So we are able to get information on the entire genome of an organism, so up to gigabases of information, whereas Sanger sequencing, you sort of aim for somewhere between sort of 1,000 to 1,500 base pairs of information. So because we have this massive chunk of DNA, gigabases of DNA that we want to sequence, often it needs to be fragmented into little pieces, and it goes through a bunch of different biochemical steps that allows us to then load our DNA. So we assay our DNA on something like a next generation sequencing flow cell, which looks a bit like this, and it's really tiny, and it goes into a massive machine, and this machine processes and reads out the information for us. So all of the DNA will anneal sort of to our flow cell that is within our sequencing machine. This sort of forms a lawn over the flow cell. And we have this concept here of bridge amplification. So this is how we multiply the molecules so that it can be accurately and sort of reliably detected by the machine. So I think the key difference, again, in the technology is that in next generation sequencing, we also use fluorescently labeled nucleotides, but the fluorescently labeled nucleotides, so the fluorophores, can be removed from these. So this no longer terminates sequencing upon addition of a fluorophore. So we can remove the fluorophore and then go along and add another base, capture what that fluorescence is, remove that, add another base, and so on and so forth. So amplification can continue. At each cycle, so at each sort of incorporation of a DNA base, a high resolution image is taken of this flow cell. And this is sort of a little cartoon of what it would look like. So at each position where a molecule might be, you can add a base, take a picture, know that it's an A, add another base, take a picture, know that this is a G, and add a base, take another picture, and it's a T, and so on and so forth. And this is sort of how this technology works. So each cycle captures one base. So each cycle, one picture, one base. And all of this is sort of done by the software contained within the machine. So again, critically, just to reiterate, same concept as Sanger sequencing. Each base fluoresces a different color. This is how we differentiate which nucleotide has been added. So key points from a bioinformatics standpoint. So I sort of mentioned that we have gigabases of information. We are not getting gigabases of DNA molecules being made or being sequenced in our DNA sequencer. We often have sort of shorter reads, so ranging between 50 to 200 nucleotides. So in this example here, you can sort of see a schematic of how we might bioinformatically process this data. So we always go through a sequencing sort of analysis step where we align all of these reads, so all of the data that we get, to a reference genome. So this reference genome is sort of a known compiled sequence. So we sort of anticipate that in the human genome at chromosome 8 at this particular position, this is going to be the reference. So this is going to be the sequence of DNA right here. And we can see where we can get information from these reads by looking at the context around it, right? So if it aligns to this region, fantastic. We know that this is where this piece of information is within the wider context of the entire genome. And often next generation sequencing then sort of uses both the information from the reads and the information of the context of where it is to give us more insight depending on the applications that we use. So for example, I think in lecture 3 you're going to be talking a little bit about DepMap. So this is sort of a resource of sequencing data as well as the cancer dependency data from a variety of different cancers. And this is an excellent sort of open source resource. If you wanted to get to grips with some coding on your own, there's lots of RNA-seq data available at DepMap. So this is, again, finding information about specific cancers using the context of our reference genome and inferring sort of details using these reads. Yeah. Cool. So another way that we might use next generation sequencing, or even long read nanopore sequencing, would be in species identification. So often we have environmental DNA. So this is bits of DNA that we sort of leave around our environment, and all animals do this, all species will be doing this. So whether or not this is little scales that are shed, or bits of skin, or hair, or fecal matter, you can extract DNA from these samples. And when we sequence these samples, we can bioinformatically identify the different species that might be occupying a certain niche or ecosystem. And then this is a really powerful, non-invasive way to sort of assay or find out how many species live in a certain area. Another sort of critical kind of real-world application of next-generation sequencing would be genetic counseling. So this is where you can use this information to help identify sort of risk for different disease development, and it's also important in treatment plans. Okay, so I appreciate that was a lot of information all at once, but if that was all too much or not really that interesting to you, this is now going to be just the absolute bare minimum that I think you will need to understand to be able to do this DNA analysis task. So, as we mentioned before, we have our 4 different DNA bases, and this is sort of a little brief example of all of the different next generation sequencing reads that you could get. So, this is a reference genome. So, in your task, your DNA analysis task, you will be provided with NGS reads and also a reference genome. So often we would apply an alignment software, and this would tell us where these reads lie within the context of each other and also the reference genome. So we can here see sort of a homology. So this is where these 3 reads align, this is where these 2 reads align, this is where this final read aligns over here. So we can also see that there are sort of little deviations in the sequences in our reference genome compared to our actual sequencing data. And we can point these out as sort of like single nucleotide changes. So here, cytosine to thymine, C to G, G to A. So your task is to write a program that will identify mutations in next generation sequencing reads. So essentially string handling and string manipulation. So we provide you the reference genome, we provide you with the sequencing reads, and critically you only have the first 200 nucleotides, which are already aligned to the start, provided to you. So there's no need to write an alignment software or anything of the sort. The task is literally to go through all of the sequencing reads, or write a program that will go through the sequencing reads, find the mutations, store this information, and then you can then go on to represent this information in an intuitive way with graphs. So the task will be on a Colab notebook. So I'm going to now try to move over to the Colab notebook. Right, so here we are. And all of this information is again repeated here. So the provided materials, what your task is, the essential functionality, so things that you need your script or your program to be able to do to get the minimum marks. And then also we have sort of more advanced functionality. So if you're trying to unlock sort of higher grades, this is kind of where you want to be putting some attention to. So it's all lined up from steps 0 to 5, and we start by importing all of our necessary modules and libraries. And then we need to download our data. So if you just hit run on this will retrieve the data from hosted on this GitHub server. So once we have that, we want to load our data into a way that can be read. So we have this function that is provided here for you. So you don't need to change anything about this function and don't need to change anything within step 2. This is literally just so that your data is nicely read in. You don't need to faff around with that, and hopefully it just makes writing the mutation identifying program a little bit easier. So yeah, so here we have sort of a printout of some of the reads. So how it's laid out, we have our read ID. So each sequencing read has a unique identifier, and we have another column which is the sequence information itself. So these are 200 nucleotide long. Write something to identify the mutations within this and store the position and etc. Cool. So very important, please put all of the code that you want to have marked into this step 3, this code space here provided. We will be marking it looking at this code space, so that's important that you include all of that there. And also when we move on to sort of step 4 and later step 5, we have a couple of test data sets that you can play around with to test that your code is able to handle errors or able to work, well, first of all, able to do the key functionality that you sort of want it to do. So here, is it working correctly, basically? This is our reference sequence. Very simply, a bunch of A's and 1 C. And then we have a wild type read, so this matches our reference, and then we have a mutant read and another mutant read here. So here the last base deviates, and here we have a deviation here at the second base. So what we want is with this reads data frame here is that if you pass this into your mutation calling function, it should return to you that you have 1 wild type read and 2 mutant reads, and it should tell you the position, so position at 200 and position 2. So yeah, that's quite critical as well. We need to remember that Python uses 0 indexing, but in biology, we don't use 0 indexing. We start from position 1 and we end at position 200 in this instance here. And then this is a great opportunity for you to as well to develop robustness to errors. So here we have some more test data, which includes things that might potentially throw an error. Yeah, this is a nice opportunity to sort of build into your code sort of this defensive programming practices. So here, truncated reads, reads that have lower cases instead of the capital that we usually associate with this format. And then also you could try writing your own test data in a similar style here to deal with unknown characters. Yeah, so that's basically that. So let me just make sure that we are back on here. All right, so I want to go over some quick example outputs. So the following examples that I'm going to give you here, so this is a completely different data set. Please don't use this to check that your outputs are correct, because it's not correct. It's a different data set to what you have been provided for your task. So at the very minimum, you want to be able to plot a graph that represents how many of the reads within the dataset provided are mutant or mutated reads containing a mutation, and how many are wild type reads, so reads that match exactly to your reference genome. You don't have to plot a bar graph. This is just the example that I've shown here. So, you know, this is what's important is that your information is legible, it's clearly presented, and just from a glance, you know, I can read and understand what you're trying to convey. So here's an example of a slightly suboptimal plot. So this is sort of the representation of where all of the mutations occur from, you know, the start of your sequence all the way up into the end here. However, the text is really small, it's incredibly crowded. I think I can guess what this is, but it's not intuitive. So you would want to move on to something perhaps a little bit more like this, where you have continuous axes. It's a little bit easier to read and understand what's going on. You want it to be well labeled, you know, in correct axis labeling, correct plot labels, and you can also include annotations within the plot if you would like to. If you wanted to represent it a little bit more like something you might encounter in sort of actual NGS literature, you could consider representing data as a heat map or any of these slightly more complicated graph types. So this is also just like another quick example of a different type of analysis that you could do to unlock higher marks. So if, let's say, you wanted to quantify not just how many bases or how many reads contain bases that are mutated, but what the mutations actually are, as well as the type of mutations these are. So just some more examples. This is really an opportunity for you to be a little bit, more free with how you want to analyze the data. It's up to you. Get creative if you'd like to. And to me, this is the fun part of coding. Cool. Yep. And as we mentioned, you can also, if that's all too easy for you, if you want to challenge yourself, if you want to try and unlock some higher grades, have a look at the advanced functionality as well. Okay, so I hope that's everything. Have fun with the task.